Chapter 14, Segment 1, pages 433 through 439. Biological rhythms. Biologic rhythms range in length from minutes to seconds or months to years. Daily rhythms have been studied the most. Many animals show daily rhythms in activity. Circadian rhythms. Most functions of any living system show an approximate 24-hour rhythm. Latin circa is about, and dies, day. Diurnal, active during the day. Most humans and most primates are diurnal, whereas most rodents, including hamsters, are nocturnal, which means you're active during dark periods. Throughout the day, our various physiological measures alter in regular, repeating fashion. This would include our hormone levels, our body temperature, and our drug sensitivity. Circadian activities show extremely close precision in the sense that they start of various activities of our day may vary in the minutes from day to day. This is present in animals via a biological clock. Circadian rhythms are generated by an endogenous clock. The circadian cycle isn't exactly 24 hours. Activity starts later each day. Free running. When an individual is maintaining its own cycle, which in the absence of external cues is not 24 hours. This period is in the individual's natural rhythm. Period is the time between two similar points of a successive cycle. This would be from sunset to sunset. The free running period is not simply reflecting external cues, because it does not precisely match the Earth's rotation. This leads us to believe that an individual has some various form of an endogenous clock. The endogenous clock runs slower in humans and hamsters, but this is not the case for all species. Normally, the internal clock is set by light. Phase shift is the shift of activity produced by a synchronizing stimulus. Entrainment is the process of shifting the biological rhythm. And a Zeitgeber, which is German for timegeber, is any cue that an individual uses to synchronize its activity with the environment. Light stimuli can entrain circadian rhythms, which leads us to believe that the endogenous clock must have inputs from visual system. Circadian rhythms allow animals to anticipate changes in the environment. The purpose of circadian rhythms is that they sync behavior and body states to changes in the environment. Our endogenous clock allows us to anticipate events and help us prep physiologically and behaviorally for the event. We can anticipate nighttime coming, so we can make preparations before nighttime actually falls. Circadian rhythms provide temporal organization for an individual's behavior. The hypothalamus houses the circadian clock. Lesions of the hypothalamus interfere with circadian rhythms. Suprachiasmatic nucleus, SCN, is a tiny subregion of the hypothalamus which serves as a biological clock. It's named for its location above the optic chiasm. Lesions that were only made in the SCN region of the hypothalamus interfere with circadian rhythms of drinking and locomotor behavior, as well as hormone secretion. Transplants prove that the SCN produces a circadian rhythm. Ralph and Minaker found a male hamster with a short, free-running activity rhythm in constant conditions of 22 hours, compared to the norm of 24 hours. Half of this male's offspring had a shorter circadian rhythm, which means this is a genetic mutation that affected the endogenous clock. Animals with 2 copies of this mutation showed a 20-hour free-running activity period. This mutation is named tau after the Greek symbol used by scientists to represent the period of rhythm. The animals entrained a normal 24-hour light-dark period just fine. In constant conditions, though, the abnormal circadian rhythm was revealed. Transplant experiments provide us with evidence that an endogenous period is produced within the SCN. Lesion in hamsters' SCN without the mutation were placed in constant conditions and their circadian activity rhythms were abolished. The hamsters then had transplants of SCN taken from fetal hamsters with 2 copies of mutant tau gene and showed that their free-running activity matched that of the donor SCN. So the period was about 20 hours rather than the original 24.05 hours. In reciprocal transplants, the endogenous rhythm following the transplant was always that of the donor SCN, which led scientists to conclude that the SCN is the source of an endogenous circadian rhythm. Transplants of fetal SCNs can drive circadian rhythms in SCN lesion hamsters, even when the donor cells are encapsulated in polymer. In these capsules, the donor SCN is unable to make synaptic connections with the surrounding hypothalamus, so its only means of communication is by releasing a chemical signal. In mammals, light information from the eyes reaches the SCN directly. The pathway that entrains circadian rhythms to light-dark cycles varies across species. Many vertebrates have photoreceptors outside the eye that are part of light entrainment. Pineal glands. In some amphibians, this is sensitive to light and helps entrain circadian rhythms to light. In birds, the pineal gland has photoreceptors that detect daylight through the skull. In mammals, cells in the eyes tell the SCN if it's light out. Retinohypothalamic pathway. Some retinal ganglion cells send their axons splitting off at the optic chiasm to synapse directly within the SCN. This is the retinohypothalamic pathway. This pathway carries information about light to the hypothalamus to entrain behavior. The retinal ganglion cells that follow this pathway do not rely on traditional photoreceptors. Instead, these cells have a special photopigment, melanopsin, that makes them sensitive to light. Even in transgenic mice that are basically blind in every sense still entrain their behavior to light because their photosensitive retinal ganglion cells still function. These ganglion cells project their axons to the SCN, but other melanopsin-containing retinal ganglion cells project to the main brain stem and inform the brain about light to control pupil diameter. Melanopsin is most sensitive to blue-range light frequencies, which explains why blue has the largest effect on human circadian systems. Circadian rhythms have been genetically dissected in flies and mice. The fruit fly, Drosophila melanogaster, shows diurnal circadian rhythms and activity. Flies with the mutation that disabled the gene period, PER, were arrhythmic when transferred to constant dim light, meaning that their internal clocks weren't running. Even slight mutations of PER could result in animals having a longer or shorter free-running period, depending on the exact change in the gene. More genes were discovered that influence the Drosophila circadian cycle. And in mammals, it was discovered that we have one or more versions of each of these genes. These various genes provide a molecular clock, which regulates each other's expression, such that their protein products wax and wane in a cycle that takes about 24 hours. Cells in the SCN produce two proteins. In Drosophila, it is clock and cycle, whereas in mammals, it is clock and BMAL1. These proteins bind to create a dimer, which is a pair of molecules joined together. The dimer binds to the cell's DNA to promote the transcription of the two genes, and the resulting per and cry, which is cryptochrome, proteins dimerize, and they inhibit expression of the clock cycle genes. Per-cry proteins either degrade or become chemically modified. The inhibition is lifted and the whole cycle starts again. The entire cycle takes about 24 hours, and it is this cycle that drives the 24-hour activity cycle of SCN cells. Each SCN neuron utilizes this to keep the time approximate, and all the neurons communicate with each other via electrical synapses. They sync their activity to produce a precise period of approximately 24 hours, which drives the circadian processes throughout the body. The same molecular clock operates in almost every cell in the body. They are all in sync as long as the SCN is intact. One possibility is that the circadian rhythm present in the body temperature, which are driven by pacemakers, may serve as a body-wide cue to entrain the molecular clock in all cells. Light passes through the fruit fly's body into the brain cells and degrades the cry protein and syncs the molecular clock. Mammals utilize the retinohypothalamic tract to deliver light information to the SCN. The cells containing melanopsin detect light and release glutamate in the SCN, which in turn trigger a chain of events in the SCN cells that promotes the production of a PER protein. When the photoperiod is shifted, the light-mediated production of PER shifts the phase of the molecular clock and thus the animal's behavior. Mutations in genes involved with the molecular clock affect circadian behavior. Mice in which both copies of the clock gene are affected show severe arithmicity in constant conditions. People who feel energized in the morning, larks, likely carry a different version of the clock gene than night owls. Different versions of the alleles of the purr gene are also associated with being a lark versus being a night owl.